Welcome, everybody. It gives me a great pleasure to welcome you to the seminar today, which is a joint, that's me, sorry, I'm going to turn off my own microphone. Joint seminar. Jointly between the Quantitative Life Sciences and CAMBAM seminar series and the STAGE strategic training and genetic epidemiology seminar series. So we have today with us Professor Magali from the Université de Grenoble Alpes and CNRS in France. I'm going to give you a little bit more background about her in a second, but first I'm going to say a little bit more about QLS, which is the Quantitative Life Sciences doctoral program at McGill. For those of you who are, there's people online, right, who are not normally here. The STAGE, this is part of the STAGE International Speaker Series. The STAGE Strategic Program in Genetic Epidemiology is training students in genetic epidemiology, not surprisingly, and the International Seminar Speaker Series is broadcast across the country of Canada and to trainees in different provinces. There's actually a regional set of nodes that are just starting, so this is actually in conjunction with STAGE Quebec, which is new. The idea here is to also promote collaborations across the country. STAGE Quebec is being supported by something called CANSI Ontario, the Canadian Statistical Sciences Institute of Ontario, which is an extra departmental unit in the Faculty of Arts and Science at the University of Toronto. It's also supported a bit by McGill, University of Montreal, Université Laval, and Grenoble, Quebec. So this seminar today is sponsored by CANSI Ontario, by the QLS CAMBAM seminar series, and we're so happy to have you here. So Professor Richard had a thesis that was actually in biology, in genetics and neurobiology from l'École Normale Supérieure de Paris. And then she went to Lyon for a while. And she was also at the BCM. BCM?
It's a team.
It's a team
of biostatistics.
Of biostatistics. Yeah. So she started in biology and moved over more towards biostatistics, and then has moved from there to Université de Grenoble and CNRS. And we are very much looking forward to your presentation today. I should say that she has said she's happy to have questions during the talk, if you have some, of course, at the end as well. For anybody who is online, feel free to put a question in the chat. I'll be monitoring the chat, or to raise your hand. I will try and also monitor that. And you can turn on your microphones to ask questions when I tell you to do so. Okay, take it away.
Thank you very much, Celia. Good morning, everyone. Thank you for being here. Thanks to all the sponsors that I cannot remember exactly who is who, but thank you, and thank you for the people online that I cannot see. Today I will speak about tumor heterogeneity, so feel free to interrupt if you have any question of clarification, and I will stop at some point of the presentation so that we can discuss. And today, if I can, okay, perfect. So I cannot switch slide. Okay, it was almost perfect. Let me.
You can't switch.
No, okay.
Oh, it worked. Okay, good.
Okay, so today I will speak. I will start with an introduction about tumor heterogeneity, and then I will show you how we can study this heterogeneity using statistical and computational methods. And to make you understand our work in the group, I use the famous painting of Claude Monet, the Nymphéa. And if you are looking at individual brush strokes, you cannot get the full painting you cannot understand the image, and this is because these individual elements interact with each other to make the painting. And this metaphor illustrates how complexity can emerge from individual elements, which is just like in a tumor. So for decades, cancer biologists have studied individual cancer cells, missing the big picture, which is that these cells interact with each other to shape the tumor behavior. So if we want to understand the tumor biology, we need to consider both individual variation and the patterns that emerge from interaction between these variations. So when I'm speaking about tumor heterogeneity today, I speak about all the differences that we can observe between tumors, as if we were looking at different paintings of Claude Monet. So this heterogeneity can occur between patients, so between tumors of different patients, but we can also observe this heterogeneity between tumors within the same patient or within the same tumor. So I will clarify what I mean. Here, when I'm speaking about heterogeneity, I'm referring to the cell composition of the tumor. So when cancer occurs, it always starts with a simple single event, which can be a mutation or a change in gene expression. And then it will shift the normal behavior of a cell towards a pathological behavior. This cell will divide, develop, and form what we call a cancerous clone. And you can have the cohabitation of different clones within a single tumor. And these cancerous cells are living in what we call an ecosystem, which is composed of different cell types, which are not always cancerous. And this is the basis of what we call tumor heterogeneity. And this heterogeneity evolves in space. So if you look at different locations in the tumor, you will see a different pattern of heterogeneity. And it also evolves with time. For instance, a simple example is the metastatic event, where cancer cells will migrate in different organs, and then you will have a development of heterogeneity of a new ecosystem. So why is it very important to study tumor heterogeneity? Well, a major challenge now in personalized medicine is to improve the predictive power of the models. And we know that tumor heterogeneity has a direct effect on the evolution of the disease and the resistance to therapy. So according to the composition of the ecosystem, you will be more or less sensitive to therapeutic treatment. And we also know that tumor heterogeneity has an indirect effect on the mathematical models that we are using to predict the evolution of the disease. It means that if you don't account for tumor heterogeneity, your models will be biased and wrong. So it's really fundamental to better understand this heterogeneity to improve our understanding of cancer biology and disease treatment. And this is where my group stands. So what we are asking, what we want to do is to use high-dimensional molecular data to better describe tumor heterogeneity and predict its effect on disease progression. So if I summarize what I have been introducing, we can say that the cellular composition of a tumor is heterogeneous in space and time, and that understanding this heterogeneity is fundamental to understand cancer outcome and survival. I can stop here. I don't know if you have any question on the introduction. Okay. Coming from a quantitative life science background, I'm sure that all of this is very clear for you. So now I will say a few words before going further. So I'm a biologist by training, how Celia was introducing. I'm not a mathematician, neither a computer scientist, but I use bioinformatics and biostatistics as tools to explore biological questions, but I'm really interested in the biological questions. And some of my explanation might lack precision or contain mistakes, so feel free to ask or correct me if required. And I'd just like to mention that it's a really fast-moving field, so staying up to date with the literature and the methods that are developed is a constant challenge. And if I'm being honest, it's a challenge that I am not able to deal with. So it's I would advise all of us to stay humble and to do our best, but it's really complicated with this field. So today, I'd like to present you 3 different projects of my group. The first one is a benchmarking of existing methods. The second one is the construction of a single cell atlas. And the last one is a project related to the spatial organization of the tumor. I don't know if we will have time to address all of these questions, so maybe I will ask you to vote at the middle of the presentation to see which subject you prefer to listen. But I'll start with the benchmarking of existing methods. So when we want to understand tumor heterogeneity, what we want to do is to quantify the cell type composition of the tumor. And this has been done for a while by cancer pathologists who are able to look at the microscope to a slice of tissue and to count and to look at different cells present. So this is very efficient. It works, but at a very high low resolution. It means that you cannot see many cell types, and it's time consuming. You cannot have a doctor at your side each time you are working with this when you are doing research. So it's a powerful manner to look at the heterogeneity, but we need to have other tools. And on the other side of all the possibilities, we have what we call single cell profiling. So I'm sure you have heard about this. Now we are able to isolate single cells and to do their molecular profile or their marker surface profile. And we can identify the cells, and this is a very good way to assess the cell type composition of a tumor. However, those techniques are very expensive and they are not used in daily routine in clinics, so we don't have many samples. So what we are doing is we are using this high-dimensional molecular profiling of bulk sample as a proxy to assess the composition of a tumor using deconvolution tools. Before going further, I'd like just to remind you what is high-dimensional molecular data. So can you raise your hand if you have never heard about methylome or transcriptome? Okay, so I think we can go fast on this. So in my group, we are mainly using methylome, and we are using the chip which contain 800,000 features by sample. And we are also using transcriptome, and for transcriptomic data, we have around 20,000 features by sample. So those are the data we are using as a proxy to infer the cell type heterogeneity of a tumor. And what we have in massive amount in clinics are bulk data. So bulk data are coming from a mixture of cells where we have extracted either the DNA or the RNA that we have profiled. So for each sample, we have a vector of feature, so either DNA or DNA methylation of RNA, and we have this for all patients.
So this is the table.
Okay.
So this is the volume of lysa that we'll be adding, NP40 and LSP, the 10x LSP.
Yes.
And this will be the final volume that we will be adding.
Got it. Thank you.
Okay. We can stop for any question if you have questions. Okay. So what we want to do is to use this data to infer the cell type heterogeneity. And we are using deconvolution. So what is deconvolution? It's a very famous approach in many different research fields to unmix a signal. So if we are looking at deconvolution from the bartender point of view, basically we have a basket of fruits. Then we do a smoothie, and what we want to do is to recollect the recipe of the smoothie using deconvolution approaches. When we are bioinformatician, what we want to do is to unmix the signal of a complex matrix. So basically, we will generate 2 different matrices. The first one, which we call the reference matrix, which is a typical molecular profile of each cell type who are present in the sample. And the second one is what we call the proportion matrix. It's the representation of the proportion of each element in each sample. And these data are representative of tumor heterogeneity. To do this, we have several kinds of methods, and we can classify them into 2 major groups. The first one are the methods that we call reference-based, for which you use known profile, molecular profile, and apply different algorithms such as regression or SVR algorithm. And those approaches are supposed to be powerful, but they are really limited by the quality of the data that you are using as reference. And in biological sample, as you may know, usually our references are very bad. So this is a limitation. And the other methods are called reference-free. And for this method, you will infer at the same time both the proportion and the reference profile. Those methods are very powerful. You get a true mathematical mixing of the signal. But the problem here is the biological interpretability of the signatures that you get, which are often a mixture of the true biological signal.
Just to make sure I understand what you're saying, with the references, what you're trying to do is just initialize what cell states or types can exist that you're trying to deconvolve to?
It's more than an initialization. It's basically you say that the true profile of the existing cell type are this one, and then you just want to infer the proportion. So it's really like a linear regression. You cannot add some fancy tools on it, but the principle is already this. You will get the beta coefficient of a regression, which are the proportion of your signal. So you won't, in those models, you won't correct this according to the result. You just start with a fixed profile. Here, you can, in this factorization method, you will initialize your problem. You might initialize it with this reference, but you will co-construct your two matrices. Then you will correct for the reference you have used.
Okay.
You have other questions? So when I started this project, it was like 7 years ago, and you had a lot of methods that were reported in the literature, and this is because it's a very famous field also in signal treatment and computer science, but with limited application in oncology, no clinical implementation. And the problem is that we were lacking objective comparative studies of this method, especially applied to the inference of tumor heterogeneity. And the difficulties of this were linked to the fact that we have bad quality of reference data sets, that if we want to benchmark method, we need to generate ground truth data, and this is not trivial, and that constructing a fair evaluation and benchmarking framework is really not that easy. So that's what we tried to do at the beginning. So this is the work of Clémentine Descamps, who was a PhD student at this time. And basically, we decided to focus on the reference-free method only on applied on DNA methylation. This is a field that Celia is familiar with. And what we did is that we constructed a benchmark framework with simulated data where we knew the true proportion matrix. And we tested different methods, non-supervised method, to infer the proportion matrix. And we computed an estimation error for all of our predictions. And what was very interesting with this first work is that we saw that all the methods that we tested were quite similar. The performances were almost the same, but what matters was really what we were doing before applying the deconvolution algorithm, meaning accounting for confounders, feature selection, estimating the number of cell types to infer. So this was our first attempt to see more clearer what was going on in the field. And then we decided that we wanted to make a more systematic benchmark, looking at different types of molecular data. So if we are coming back to our smoothie, the idea was to use multimodal data to assess the deconvolution efficiency of the method. So for a smoothie, it's like you wanted to test if your test, to try if the test was better to estimate the recipe or the color of a smoothie. So for instance, if you have kiwi in your smoothie, maybe you will use the color to estimate what is the initial recipe. But if you have lemon, you might want to use the test because it will be easier to see the acidity. So the idea for us is to use both DNA methylation and gene expression, so RNA, to do the deconvolution. And this was the work of Elise Amblard, a postdoc in the team, who was very, very brave, because when she arrived in the lab 3 years ago, she was supposed to develop a new method. And at the end, she spent 3 years to develop a benchmark, and she didn't have time to develop a method. But this is a very courageous work. So She built an entire framework with different kind of data set, in vitro data set, in vivo and in silico simulation on different kind of tissue with two important steps. So the first one was to apply deconvolution, and we tested like around 20 different algorithms. And the second one was a very complex ranking pipeline so that we can assess if we see small differences in the performances, would that mean something? So we built an ad hoc statistical testing for this. And she looked at different metrics of performances, so some error metrics, but also some correlation. She looked at the stability of the algorithm. So are the performances reproducible from one data set to another one? And she looked also at the scalability of the method, meaning the computing time. So with this, we provided this kind of data where we can see the different method and if they behave differently than the other one. We also provided what we call atomic score for each method, meaning we can look at the different behavior of a method depending on if we are looking at the error or the stability or the correlation. And this is important for biologists because they might want to use a different method according to the questions they are asking. And what we also provide is a comparison of the efficiency of the method depending on the molecular data that we are using. And what we saw is that basically the method the performances are comparable given the molecular data used, which was a new result. And we also saw that the best method was a very, very simple robust linear regression. So you have many methods that are developed several per year with now a very fancy machine learning algorithm, but still at the end, it's okay to start with a very simple method that you understand. And to finish with this, what we also did with the Slim Karkar, a postdoc of the team, is to develop a web application to help clinicians to use methods. So here we focus on unsupervised method. And on this application, you can put your data set. We provide a deconvolution module with several algorithms and a biological interpretation module that will help the biologists to understand the components they have inferred. So that's it for this part, and I would like to finish with the fact that all of this was made possible thanks to collective intelligence and collaborative work. We organize several scientific competitions, which is another word for data challenge, dedicated to this topic. So what is a data challenge? I don't know if you already play this kind of game, but basically it's a problem to solve. Participants that will try to solve the problem and an evaluation framework. And for us, organizing a data challenge is a very long process. Like it take us a year to organize a challenge. Then we have the event, and we have a phase of post-analysis and publication, which is almost 2 years or so. So we organize 4 events dedicated to deconvolution, and we publish collectively the result of the challenges, and it was very important for us. So I advertise this approach because for me, at least, it led me to have new collaboration. It led to scientific publications that are collective and not individual, which is important, I think. It also helps to raise money to generate ground truth data set, which is not always easy to do. And it also engages a community around a common scientific question, which is also important. And this is the last event that we organized. It was last year. It was in the Alpes. We had a lot of cheese, and it was very nice. Okay, so I'll finish with this part. So as a summary, I would say that the cellular composition of the tumor ecosystem can be quantified using bioinformatic approaches. And it's always important to explore existing tools and methods before developing something new, even if it takes you 3 years to do so. Often simple algorithms perform best. Complexity is not always better. And collaborative science is essential for benchmarking and improving algorithms. I can take a few questions if you have some at this point.
I do have one.
Yes.
Did you look at performance of rare cell types?
Yes. And what we saw is that RNR still perform best. So it's the best method, whatever the abundance of a cell type. But we were a bit puzzled because the rare cell types are the immune cell types and that are also the most correlated ones. So we don't know if we are looking to the problem of correlation between cell type or sparsity, and we try to force sparsity of more different cell type, for instance, fibroblast, and it was not that difficult to estimate. So I think what we usually think of the problem of sparsity in the sample is really linked to the nature, the correlated nature of those immune cell types. So that was quite a discovery for us.
That's going to be really interesting, right? Because some of these very, very rare cell types are thought to be so very crucial in certain situations.
Yeah, no, we were quite excited about this, but still, this correlation exists in the biological system, and it would be a problem, but it can help us to think differently about this problem.
But there would be a problem to get reference data, presumably.
Yeah, of course.
Yeah. Any other questions? And questions, are there people? I don't know if they can hear me over here. Those online, do you have any questions?
I have one. I'm curious about how you might approach these sorts of evaluations and deconvolutions if you're looking at like cell types or annotations that are not like super discrete. Because like sometimes you can kind of follow in between two different points on like a scale of differentiation or something like that.
Yeah, no, this is for sure a very important problem, and we didn't come up with good metrics to assess this. What the best we can do so far is to really diversify the dataset that we are using and the ground truth model, meaning that for in silico simulation, we have the real ground truth. But we combine this with in vitro data and in vivo data, for which the ground truth is very bad, but it's complementary. So, for instance, we have an in vivo data set where the ground truth was observation of a pathologist on a microscope. So, for this, you have the feeling of the pathologist. You will say quantify with some plus and minus if a cell type is present or not. But I think this is more close to the true continuum of differentiation of a cell type than what we call as a static reference. So I think that's the way we can deal with this so far, but it's definitely a problem when we assess the performances of the model.
So it was pretty clear that the unsupervised methods performed worse. It was kind of like almost like a straight line going across.
Yeah.
I was wondering if there's a particular benefit to using them. Like, there must be a reason why people are diving into unsupervised methods.
Yes, if you don't know what you are looking for, if you don't have references, or if you are looking to unknown cell types or unknown properties that you are not sure you can characterize yet, it's really good to go with this unsupervised method. So you are not restricted to the input that you put. And some recent methods are combining both approaches. So basically, you can use references for some of the components you will deconvolve, and you can leave a blank component that will be inferred by the model. So I think this is a good way to do deconvolution. And but in the ideal world, we will improve our references, and we won't need any unsupervised approach anymore. I'm not sure we are at this point yet. Yes?
So you benchmarked a bunch of algorithms against each other. How did you rank them and score them?
We don't want to rank them because our message is really to give a recommendation on which algorithm you will use. But you're right, we did this like p-value, ad hoc p-value to rank them. So we did this using permutation test. So it's like an empirical p-value that we computed for each metric. And we did that because often you see in the benchmark that a very small increment in some metrics will lead to a different ranking, and we did not want to do this. So the idea was not to show that a method was the best, but more to discriminate like bags of methods that were behaving similarly. And another aim was also to see if methods that were ranked similarly were based on the same mathematical algorithm, which is not always the case. So basically, you have very bad methods. You have some methods that are doing not so bad, and you have some methods that are better, like robust linear regression.
I have a question online. I think which I quite related actually. Would the speaker be able to clarify the benchmark method or score to assess performance of the deconvolution methods? Was simulated data involved?
Yes. So we had different simulation framework, which we did improve since the last paper. So now we are using simulation based on collinearity between features, which we were not doing at the time. So maybe we had just one for the reviewer. But so we simulate the data using a pure reference profile that we mix together, adding some biological noise and technical noise. We have in vitro data set where we at the bench with a pipette mix pure cell types, and in vivo data set. And then we have several metrics to measure the error, which are based on mean square error, mean absolute error, and different correlation metrics. Does it answer the question? Yeah,
I think so.
okay. Okay. So we move to the next part. So I think we need to decide which will be the next topic. I can speak about the construction of a single cell RNA-seq atlas that we did in order to improve the references that we were doing in the deconvolution. Or I can speak, so you probably don't see it well here, but the second option is to speak about spatial organization of the tumor. So I don't know if you have a preference. I think if I do both, I will be too quick and it won't be interesting. You have a preference?
Yeah, who's in favor of number three?
Spatial organization. Oh. Okay. I really like the atlas, but I will say a few words about it. Only one slide. So the idea here was to use a public data set, single cell RNA-seq data set to improve the references. So all the methods I have shown you before are called type 1 or first generation method, and they are based on bulk references. And there are a bunch of methods that were developed more recently based on single cell data references. So we wanted first to use atlases that were built and published. And we realized when we tried to download it that they were not accessible. So we had to build our own atlases. And that's the work of Lucie Lamotte, an engineer in the team. It's a very nice work, but it's not for today. I will just present this because this is important for the last part. So our work really focuses on one typical cancer type, which is a pancreatic adenocarcinoma. This cancer is very bad because the prognosis is terrible. And this is due to the fact that it's often a cancer that is late diagnosis, but also we have a poor therapeutic option related to the fact that it's a very heterogeneous, highly heterogeneous cancer. So here you have the representation of a normal pancreas, and when cancer arises, you have few cancerous cells and very dense stroma, so microenvironment that can be heterogeneous. And we know that there are two types of cancer cells that we call classic and basal. The second one is more aggressive, and it's a dedifferentiated cell type. And we are working in collaboration with Jérôme, who is a pathologist in Paris, expert of this pancreatic adenocarcinoma. So all that the work I will show you in the last part is based on samples that were provided by Jérôme, and it's really related to pragmatic concept. Okay, so I will move on. The last, your favorite part. Okay, so what we see in this pancreatic cancer is that we have a different kind of spatial structure of the tumor. We have simple cases where you have only one type of cancer cells. So you have the type blue and the type orange. So those are the slides illustrating this configuration. So I don't have, okay, those are for distinct compartments. So I don't have illustration for those two. So this is the first case. The second case is that you have a distinct compartment of orange and blue in the same tumor. And this is what you can observe here by staining. So on the same tissue, on the same tumor, you will have either patch of orange cells or patches of blue cells. In some cases, you have these homogeneous patches, but also patches of cohabitation between orange and blue. And you can also have some cases where you have just the coexistence of the two cancer types. And we have no idea of what is driving this configuration. And as you don't have the same aggressivity regarding the type of cancer cells that is present in the tumor, for us, it's very important to understand what are the drivers of this heterogeneity. So to do this, we generated spatial transcriptomic data on 12 samples because we were very rich. No, this is a very expensive technique, so it's one of the difficulties that you don't have many samples. And this is a project of Luis Martin Peña, who is a PhD student in my team. So we use this spatial transcriptomic technique. So do you know, I guess if you wanted to hear about this project, you know the technique, but who has never heard about spatial transcriptomic? Okay, so move on. So it's, you have several techniques, several degrees of resolution. Some are based on molecular data, some are based on fluorescence. In our case, we are using 10x vision, which is the most common technique based on molecular profiling of RNA-seq data. And the resolution of this technique is not so big, and I will show you later why is it important. But at the end, we have an expression matrix of genes per spot and coordinates. In our case, we have only X and Y coordinates. So what are the challenges associated with this kind of data? So it's really a fancy technique. Many methods are developed to solve the challenges, and I will focus on the few that are interesting for us. So first challenge is to identify what we call a spatially variable gene. So a classical question that we ask when we are dealing with the expression data is differential analysis. We have sample A, sample B, and we want to know which genes are differentially expressed. So here, it's the same question, but you add the spatial layer. So you want to see a differential spatial pattern. So this is the first challenge, and you have some method like SparkX, which are doing this very well. Another challenge is, and this is of interest to me, a deconvolution challenge, because in this spot, you don't have one cell, you have several cells, sometimes 10, 20, 30 cells. And it's very important to be able to address this question to better interpret the biological observations that we make. And some dedicated methods have been developed to address this particularly. Last challenges are to identify relevant spatial domains. So you have your data, you can do unsupervised clustering to correlate the spot to each other, and then identify spatial organization and to see how the spots are interacting between each other to infer some hypothesis on cell communication and on biological driver of the spatial organization. So this was a small introduction, it's not our work. but I hope it will help you to better understand what we are doing. So if we are coming back to our sample, so we generated 12 samples, and right away we lost 4 of them because they are not supposed where they were supposed to be. So basically we couldn't identify any cancer cells on those samples. So they are very nice, but they are useless to us. So we focus on the remaining 8. And this is a very simple approach to identify some or to label some spot, which is called U-cell. So I don't know if you have heard about this, but basically you use some marker genes to have a probability of identity for each spot. So we did that as a preliminary analysis to classify our sample. So we had identified 4 samples that were labeled like pure classic, 1 sample pure basal, and 3 samples with a mixture of the 2 cell types, 1 with distinct compartments and 1 with more mixed compartments. As you see, it's not as easy as it is with simple labels to characterize these images. But what was reassuring for us is that it correlated with the pathological annotation. So our collaborator, the anatomopathologist, looked at the data and selected those samples for those reasons. And we were able to match the observation. So then we wanted to go further and not only add raw labeling of some spot according to a few gene markers, but we wanted to perform deconvolution of each spot to really understand what was the composition of the microenvironment in this sample. So to do this, we use our wonderful single cell atlas that you don't know about. So it's really recent data, but we start to be confident with it, and we have like a million cells that are annotated, and we have 20 different cell types. So we use this to perform the convolution of the spot, and here was the first nightmare of the project. So to do the convolution again, you have like 25, 30 methods that are all based on different algorithms, and it's really complicated to decide which method to use. So we tested the most famous one, which didn't work in our end, and we called some colleague who told us that it was the same for us. For them, so basically we readjust which method we wanted to use, and we ended up with this spotlight method, which is quite old, like I think it was 5 years ago, maybe more. It's a paper I sent you for the journal club. But it's working quite well, and it's fast enough so that we can test many things. If you want, we can come back to the principle of the method more in detail later or during the lunch, but roughly, this is a combination of non-negative matrix factorization to identify topics that are functionally driving the cell type identity. And then it's regressions to identify the composition of the cell of the spot. So with this, we were able to have a precise annotation for all of these cell types of each spot of our sample. So the pictures are not pretty, or even they are ugly, but it's really ongoing work that I'm sharing with you now. So what you see or don't see is that you have small pie charts for each spot that are indicating what is the cellular composition. And what was good for us is that we were able to see the patterns that were identified in the previous analysis. For here, for instance, here you see that you have a red block here and here, which are corresponding to what we annotated with U-cell. And here you have a yellow block. So trust me when I say it, because it's impossible to see what is the picture. And what we are doing now with this is that we also tested another deconvolution algorithm that is working well, which is called SPADE. It's a more recent one, which is also based on unsupervised clustering and domain identification. And we want to do consensus annotation using different methods to estimate the best as possible the cell type organization. I won't go into the detail of this, but the consensus approach basically is to try to take the best of each method instead of choosing one or cherry picking one. You have a criteria to mix several outcomes, and we can do this with the convolution by using the error of reconstruction of the initial matrix, and we can weight the result of different methods. And this is what we are working on now. So now I will stop showing you results because this is all questions and prospect and hypothesis that I want to share with you. So the first question that we want to ask with this data now is what is different between these cancer cells? So we identify some domains that are related to either basal cells or classic cells. And what we want to do is to do differential analysis to identify genes that are differentially regulated between these clusters that could explain that they are located nearby another type of cells. So we will perform this differential analysis using an approach that is based on nonlinear analysis using Gaussian kernels. And we will also would like to try to perform some explainable machine learning, but this is so far it's a dream, so I don't know if it will work. But the idea is to construct a gene regulatory network of each cell type, a cancerous cell type, and then to try to predict the identity of a cell type given its spatial location. And from this, we can use sharp value to see which features are important for the prediction of the identity. Theoretically, it works. We'll see in reality if we get the funding we ask for. Then the second question would be the characterization of the spatial niches of this domain. So here I put in gray what we can call the neighborhood of cancer patch. And we would like to detect if we see differences in the structure and composition of these niches. And so recent method proposed to identify niches based on a graph-based approach. And with colleague expert in stochastic modeling of the graph, we would like to try this. And again, this is really prospective. We didn't start it yet. And the third question we'd like to answer, if we are able to define the niches, is that can we take advantage of the million cells that we have identified to predict their spatial location from non-spatial data. So basically, we have RNA-seq data for these single cells. And using the definition of the niches we have done from spatial transcriptomic data, can we do this prediction? And here the challenge is that we have very few data sets for spatial transcriptomic. So if we want to do this, we need to be able to do data augmentation to be able to train a model on this data. And then the classifier model that we will use, it can be anything, it can be random forest, it can be very fancy deep learning method. But this is not important. What is important here is to do data augmentation, and this is really not tricky. But we'll try. We will see if we succeed. Okay, so I think I will finish with this. So as a summary, I have shown you that the heterogeneity evolves via mutation, clonal dynamic, and cell plasticity, and that this plasticity can enable transition in response to the microenvironment. And if we want to understand this, we can use spatial transcriptomic, which can reveal niches, gradients, and cell-cell interaction. And that machine learning approaches could be powerful to understand all of this and link cell composition, spatial behavior, and functional state, but it's hypothetical, so we need to test it and to demonstrate it. I will give a brief summary of the two parts we discussed. So the first part was about benchmarking the convolution algorithm. And with our work, we highlight the importance of simple validated method and collaborative science. And the last part was about spatial analysis. I would like to emphasize that we are really dedicated to reproducibility and good practices. It means that we are using computing clusters containers, workflow manager, and versioning. And if you want to discuss it, I will be very happy to do so. And I will finish by thanking all the people that were involved in this work, so the member of the group, and especially Elise, Clémentine, Lucie, Louis, who is not on the picture because he arrived recently. And all of those people also have been involved in the scientific competitions that we have run and contributed to the work. All the collaborators. And I'd like to finish by saying that our group recently moved to a computer science lab, so we will be closer than ever to a computer scientist. So maybe we can make this machine learning approach work. We'll see. And I will come back here for a 6-month research stay from July to December. So I will be happy to discuss with you at this time also. And thank you for your attention.
I was curious about in the last part about the biological significance, the importance of the niche that you identify. So there are clusters of cells that have particular properties that you identify, but then what do you make of them? How do you use that for better understanding the system?
That's a very good question. First, we will be happy to describe it because it has not been done. So we have this 8 samples, but we have also samples from the literature. So it would be nice to join all of this and to see if individuals will share common niches or not, and how it's organized within the tumor. And then you can try to make prediction and correlation between the subtypes of cancer cells and the related microenvironment niches. So if I want to be more clear, I would say something about immunology. So we, in pancreatic cancer, we have not a clear idea of what is going on. So you have contradicting publication about specific cell types that are present in the tumor that might have pro or anti-tumorigenic effect. And so with these niches, I hope we will be able to have understanding into this mess.
And just to clarify, so that these niches are based on the composition of the cell types, but also on the when you say the microenvironment, you have other variables that characterize.
So it's the cell type, the microenvironment. It's cell type. Okay. So we could have add some other variables, but we don't have them.
When you were looking at the consensus deconvolutions of cell types, I'm wondering if you have any ideas about what to do with the spots or the cells that are not confident, that you get conflicting answers from different groups, and kind of how to either ignore or incorporate that uncertainty into the analysis.
At some point, you have to make some choices, and it depends on the question you want to ask. Some methods are using thresholding. For instance, if you have a spotlight, we say if you are lower than I don't know 10%, you are not considered, or not you, but the cell type. So I think this might help you to get confident, but you might lose some information on rare cell types. So it depends on the question. And again, on the consensus, you can give some weight related to the uncertainty. Not sure how we'll do that, but I think it's a good start.
I just have a question about the one of your future plans to compare like the differential expression between the two like tumor clusters. I was just wondering what data would you be comparing, because it's like the spatial transcriptomic data is the one that gives you the 2 different delineations, but it's like a bit more reductive than the single cell data, because I guess there's not as much information.
Yeah, you lose information, but we will use an approach that has been developed for single cell data, and that we will try to adapt for spatial transcriptomic with our colleague. I have a slide here or not. Okay. Yes, it's here. So it's a a method based on kernels that so basically here you have two genes of interest that you want to test, but you can also compare the full distribution of gene expression between two cells, and you do a kernel-based transformation of your data, and then you look at the discriminant axis. And with this approach, you can identify differences that are not based on the main mode, like for example, the mean. And this method has been proven to be efficient for single cell RNA-seq data, where you can detect, for example, variation in the zero proportion of the cells, or some modes that are distribution cues or stuff like this. So I haven't applied it yet on spatial transcriptomic data, so I don't know if it would work, but at least it has a potential to overcome the difficulties.
I have a couple of questions here. What method did you use to assign cell labels to clusters generated from the Visium data? Was it manual or a probabilistic model? And if it was manual, how many markers did you use to define a cell type?
So in the single cell atlas, I think the question is this because
Yeah.
so this is the slide that we have here. We used a very complex framework, and we made a curation of the literature to collect as many markers as possible, and then we did a hierarchical annotation. I can show it here. So we have this complex framework where first we annotate very basic cell types such as epithelium or stroma. And then we go down into the complexity with another set of markers. And we use several methods of annotation. So the first one was a pseudo-bulk single sample GSEA. So it's a gene set enrichment analysis. And we also used SC type, which is a method which is based on rank without statistical estimation. And we are finally using a consensus approach because we like to do consensus. So depending on the branch history of our labels, we can validate an annotation if the two approaches are coherent or not. So that's complicated, but we think it's a good way. I don't know if it's a good way, but it's the best way we came up to annotate.
I have one more here. Do you expect any difficulties in classifying niches for different tumor cell types using 2D data? And do you have any option for serial sectioning to get 3D representations?
Yes, I expect difficulties, and no, I don't have any options for 3D sectioning. So it's
okay. And a new one just came in.
Yeah, okay.
Because I know we're running out of time. Can you comment on the role of karyotype changes in cancer and whether the methods are actually sensitive to the karyotype changes? As in, do the cell types actually change themselves as the cancer evolves?
Yeah, we have it's not related exactly to karyotype, but we are looking at the genetic mutation, and we are trying to link to statistically associate microenvironment composition and genetic mutation of subclones. So we are looking at this also.
I think we're going to stop here because you guys all have a chance to ask more questions. So I would like to thank our speaker for a really lovely presentation. Thank you very much. Thank you to all those of you who online who joined in, and please join the Quantitative Life Sciences seminar again, and also the International Speaker Series of the STAGE training program whenever you can. Goodbye.
Thank you.